This is the recorded lecture for Chapter 21 on the Kingdom Protista. We covered some of this in class, but I'm just going to go back and review. Remember with the Kingdom Protista that we're now into... Okay, well, first of all, remember that there are 3 domains of living things. We covered the domains bacteria and archaea in Chapter 20. Now, for the rest of the course, pretty much, for the rest of this semester, we will be covering organisms that are in the domain eukarya. That means that they are composed of at least one cell, and that cell has a nucleus and organelles. The domain eukarya, the cells are larger than in the other 2 domains. And we're going to start the domain eukarya with the Kingdom Protista. And the Kingdom Protista consists primarily of protozoans and algae. So there are 6 supergroups that the protists are classified into. There's the excavata, the chromalveolata. And we covered both of these, the excavata and the chromalveolata. We covered both of these already. And I'm going to focus more on the last 4, which are the Rosaria, Archaeoplastida, Amoebozoa, and Opisthokonta. Remember that the Protista are classified in the domain eukarya. That means that their cells have a nucleus. And the Kingdom Protista. As far as the Kingdom Protista, it consists of algae, which are photosynthetic, and protozoans, which are heterotrophic. And some protozoans, and remember, heterotrophic means they ingest their food. They don't make their own food. And some protozoans are mixotrophic. We already talked about one called the Euglena, which we will see in lab tomorrow. For the Protista, asexual reproduction is the most common way for reproduction. It is basically cloning. Now, as far as their nutrition, the Protista can be photoautotrophs. And for the most part, that means they're photosynthetic, and photosynthesis produces oxygen and releases it to the atmosphere. So they are important for adding oxygen to our atmosphere, and they are a major component of plankton, which are organisms suspended in the water. The Protista are classified into the 6 supergroups. I had you guys copy this, so the information on table 21.1 is very important. It lists the supergroups and the major members of those groups. If you need to make flashcards to learn these, whatever you need to do, but this is information you need to know by the first exam by heart. Okay, so we have the excavates or excavata, and the main examples we went over were the euglenozoans or the euglena. They are the mixotrophs that are capable of photosynthesis, but they also have a flagella, they swim, and they're capable of ingesting food. They're the ones that have a red eye spot, and they're attracted to light. But then we talked about diplomonads and parabasilids, and both of our examples that we went over were parasites. Then the chromalveolata, which are really divided into the alveolates and the straminophiles. So the alveolates are the first 3 that are listed here. Dinoflagellates, which, if you'll remember, they are the cause of red tide. Apicomplexans, and one of those is the parasite that causes malaria. And ciliates, and one of those is the paramecium that we're going to learn about in lab, that we're going to see swimming around under the microscope. Now, the groups in the second and third line here are straminophiles. So they are the diatoms, golden brown algae, and water molds. All right, next we have the supergroup Rosaria, which consists of the cercozoans, foraminiferans, and radiolarians. The Archaeoplastida, which consists of the red algae and green algae, and then the plant kingdom. The Amoebozoa, which consists of amoeboids and slime molds, and the amoeboids have the main example of the amoeboids is what we call amoeba, and we will see amoeba in lab tomorrow as well. The Opisthokonts, or the supergroup Opisthokonta, contains choanoflagellates, and then also the kingdom fungi and the kingdom animalia. For the excavates, if you remember, I just want to focus on the examples. There are diplomonads, parabasalids, and euglenozoans. So the diplomonads, primary example is the Giardia lamblia, which is a parasite that causes severe diarrhea, and it is contracted from contaminated water. Then there are the parabasalids, and the example that we learned was in class was the Trichomonas vaginalis, which causes an STD. So both of those, and here's your Giardia under an electron microscope. Both of these, the Giardia is your diplomonad, and the Trichomonas vaginalis is the parabasalid. And then we have our euglenozoans, or euglena. And this is what our euglena look like. Again, we're going to see these under the microscope in lab tomorrow. They came in and they are living, and they're swimming, and they're active, and they'll be fun to study. All right. Within the euglenozoans, there is also one that I want to mention to you. It's a type of protozoan called a kinetoplastid. The name comes from the fact that they have a kinetoplast, which is a large mass of DNA in the mitochondria. But the primary example is the Trypanosoma genus. One of the Trypanosoma organisms is called Trypanosoma brucei, and it causes African sleeping sickness. Its host is the tsetse fly, which transmits the protozoan parasite to humans. And so it has two hosts. It has the tsetse fly and humans. And this is what it looks like under the microscope, along with red blood cells. So this is a blood smear of someone who is infected with African sleeping sickness with the Trypanosoma brucei parasite. So this is another example of an organism that's found within the euglenozoans, and euglenozoans are found within the supergroup Excavata. Okay, supergroup Chromalveolata, which we already again learned about in class, contains the alveolates and the straminophiles. The alveolates include the dinoflagellates, the apicomplexans, and the ciliates. And then the straminophiles include the diatoms, golden brown algae, and oomycetes, which are the water molds. I'm going to skip through some of this because we did go over it in class. And that was where we left off in class. So now we're going to talk about the supergroup Rosaria, which is composed of cercozoans, foraminiferans, and radiolarians. Now, one thing that they do have are false feet, or these extensions called pseudopods. When we learn about the amoeba, they also have pseudopods, but the difference is the Rosaria have pseudopods, but they also have a shell, and where amoeba do not. The cercozoans are really just discussed as the fact that they can live in an endosymbiotic relationship. with cyanobacteria. In fact, it's the cyanobacteria that live endosymbiotically with the cercozoa. They live inside of them. Okay, cyanobacteria are photosynthetic bacteria, so sometimes they will live inside cercozoans. And that's pretty much, this shows you that some of these, this sausage shaped, this structure right here is your cyanobacteria, and then the entire organism is the cercozoan, which is your example of from the supergroup Rosaria. But we really want to focus on the supergroup Rosaria. We really want to focus on the foraminiferans and the radiolarians. Both of these contain pseudopods, and they both contain a skeleton known as a test. The difference is the composition of their shell or skeleton. Foraminiferans are composed of calcium carbonate, which is basically a chalky type of substance. The fossils or deposits of the formed made by the shells of the foraminiferans have formed the White Cliffs of Dover, which you can Google. The radiolarians also have a test, but it is composed of silicon, so it's more of a glassy type test rather than a chalky. These are some images. Down here are your I'm thinking for a minute. Okay, well, first of all, this is the White Cliffs of Dover. These deposits, these white chalky deposits, were made by foraminiferans. And in this picture to the top left, you can see pseudopods extending out of the test of a foraminiferan. And then you can see these are radiolarians, I believe, and I know those shells look white and chalky, but I'm pretty sure those are radiolarians. Let me see. Yeah, they are. They're radiolarians because it says the spiny white skeleton of silica. So these down here at the bottom have a glassy silicon-based test. And these are the radiolarians, and these are actually just the shells of the radiolarians, or the test of the radiolarians. The radiolarians are no longer there, but their pseudopods extend out of the little holes in the test. So you can see the radiolarians here. You can see the White Cliffs of Dover that are produced by the foraminiferans, and then up in the top left you can see a living foraminiferan. Now we're going to move on to the supergroup Archaeoplastida, which contains also the kingdom Plantae. So these supergroups sometimes will have other kingdoms as members of the supergroup. And so the Archaeoplastida is the supergroup that includes algae and plants, land plants. So we have red algae, which are the multicellular seaweeds. The economic importance of the red algae is that they're used to produce agar, which we have used in class. We used agar or agarose gel to make the nutrient medium, growth medium for our bacteria that we grew in the Petri dishes. Agar is also used as capsules for drugs. If you take any type of medication in a capsule, that capsule probably contains agar from seaweed. It's used for dental impressions, cosmetics, bacterial culture medium. Sometimes that's used in a Petri dish and sometimes in a test tube. Electrophoresis, which is used for separating different size pieces of DNA and doing DNA fingerprinting. And food prep. And we also have carrageenan, which is an emulsifying agent used in the production of chocolate and cosmetics. And then the reddish-black wrappings around sushi rolls consist of porphyra blades, which is a type of red algae. So here's a picture of red algae. And then green algae. There are many, many different types of green algae. They can be unicellular and they can be multicellular. They can be filamentous and they can be colonial. So Chlamydomonas is a unicellular green algae. This is a black and white electron micrograph of it. And this is the life cycle of the Chlamydomonas. We won't go into the details of that, but it does have a good picture here. It's got 2 flagella, and it's green, so it does carry out photosynthesis, but it can move. So Volvox is one that we will see. We have a slide, a preserved slide that we'll look at in lab. Volvox is a colonial green algae. That means it's a colony of independent cells, and it's like a hollow sphere, kind of like a basketball. So all of these balls are colonies of independent algae cells, and the whole thing is called a volvox. So that's a colonial green algae. Then we have a multicellular green algae called sea lettuce, and here's a picture of that, ulva. And then there's Spirogyra, which we will also see under the microscope. Spirogyra is a filamentous green algae. And here's Chara, which is multicellular. And I will cover the last 2 groups in the next recording.